Dear learners, I am Ramya, working as Assistant Professor in the Department of Biotechnology, St. Joseph's College of Arts and Science for Women, Hosur. We are going to learn about growth and multiplication of bacteria. Bacterial growth can be defined as increase of all the chemical components of the cell. It makes the population. Most bacteria divides by binary fission, in which bacteria undergo cell division to produce two daughter cells identical to parent cell. Bacteria can be grown equated to cell number. One bacterium divides into two, and 2 produces 4, and then 8, and so on. The growth rate of bacterium is therefore measured by measuring the change in bacterial number per unit time, called as generation time. Generation time is a time required for the bacterium to give rise to two daughter cells under optimum temperature. The generation time for most of the pathogenic bacteria, such as example Escherichia coli, is about 20 minutes. The generation time is different for different bacteria. For example, Mycobacterium tuberculosis, it takes 20 hours, and Mycobacterium leprae takes 20 days. The bacterium replicates and multiplies rapidly, producing millions of cells within 24 hours. For example, E. coli in about 7 hours can undergo 20 generations and produces 1 million cells. This is how the generation time of bacteria takes place. A single bacterial cell is inoculated in an agar plate, which grows rise into visible bacterial colony in the generation time of 12 hours. Bacterial count. Microbial concentration can be measured in terms of cell concentration and biomass concentration. Total count. This denotes the total number of bacteria in the sample, irrespective of whether they are living or dead. Viable count. It measures only the living bacteria. It can be obtained by dilution or plating method. In dilution method, several tubes with liquid culture media are incubated with varying dilution of sample, and viable count is calculated from a number tubes showing bacterial growth. In a plate method, sample is diluted and small volume of it is spread on the surface of an agar plate. Bacteria can be multiplied by binary fission process. Conceptually, this is a simple process. A cell just needs to grow to twice its starting size and then split into two. To remain viable and competitive, the bacteria must divide at the right time in the right place and must provide each offspring with a complete copy of its essential genetic material. Bacterial cell division may allow for a design of new chemical or novel antibiotics that specifically targets and interferes with cell division in bacteria. This is how the cell division takes place. One bacterial cell divides into two daughter cells. It is elongated and cell plate forms in between and produces two daughter cells. Microbial growth and nutrition. What are all the things required for growth and nutrition of a microorganism? The microbial cell is made up of several elements such as carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, calcium, magnesium, and iron. These are also known as macro elements or microelements because these elements are required in high amount by microbes. Apart from these, other macronutrients are found to have several biological functions. For example, potassium ions involved in the activity of several enzymes. Calcium is an important element of bacterial endospore. Magnesium involved as cofactor of different enzymes. Several other elements are also required by the microbes on a small level. That is what we saw, that is microelements. It includes manganese, zinc, cobalt, molybdenum, nickel, and copper. This does a biological function in several ways. For example, zinc is present at the active site of several enzymes. Manganese is involved in catalysis of the transfer of phosphate group. Molybdenum is essential for nitrogen fixation. The requirement of all these chemicals, that is carbon, hydrogen, oxygen, and electron. Organic molecules are essential for microorganisms, and a major constituent of these organic molecules is carbon. Hydrogen and oxygen are also found in the organic molecules. Electrons have two main functions, which are the movement of electrons through electron transport chains and during oxidation reduction reaction. can provide energy for use in cellular work, and electrons are also needed to reduce molecules during biosynthesis. The requirement for nitrogen, phosphorus, and sulfur. Nitrogen plays an important role in synthesis of amino acids, purines, pyrimidines, some carbohydrates, and lipids. Many microorganisms can use nitrogen in amino acids. Phosphorus present in nucleic acids, phospholipids, nucleotides like ATP, several cofactors, some proteins, and other cell components. Sulfur is needed for the synthesis of substances like amino acid cysteine and methionine, some carbohydrates, biotin, and thiamine. Organisms are capable of producing enzymes required for biochemical pathways by the presence of nutrients. Several organisms that lack specific enzymes required by microbes. Therefore, they must contain these constituents or their precursors from the environment. Organic compounds that are essential cell components or precursors can be synthesized by the organisms are called growth factors. There are three major types of growth factors such as amino acids, purines, and pyrimidines, and vitamins. Some microorganisms require many vitamins. For example, Enterococcus faecalis needs 8 different vitamins for growth. Other growth factors such as heme from hemoglobin or cytochromes is required for Haemophilus influenzae, and some mycoplasmas need cholesterol. What are all the factors affects this growth of bacteria? Maybe a temperature, pH, moist content, available nutrients, and the characteristic of other organisms present. Bacteria, on the basis of oxygen requirements, it is classified as aerobic and anaerobic. Aerobic, there are few types in it. One is obligate aerobes, which can grow in the presence of oxygen. Example, Pseudomonas aeruginosa. Facultative aerobes, which are ordinary aerobes but can also grow without oxygen. Example, Escherichia coli. Microaerophilic bacteria that can grow in the presence of low oxygen, in the presence of low, that is 4% concentration of carbon dioxide. Example, Campylobacter jejuni. Some fermentative organisms, example Lactobacillus plantarum, are aerotolerant but do not contain the enzyme catalase or superoxide dismutase. Oxygen is not reduced and therefore hydrogen peroxide and nascent oxygen are not produced. Anaerobic bacteria. Obligate anaerobes are the bacteria that can grow only in the absence of oxygen. Example, Clostridium botulinum, Clostridium tetani, etc. These bacteria lack superoxide dismutase and catalase, hence oxygen is lethal to these organisms. Carbon dioxide. The organisms that require higher amount of carbon dioxide for their growth are called capnophilic bacteria, 5 to 10% and 15%. In candle jar, 3% carbon dioxide can be achieved. Example of such bacteria includes Haemophilus influenzae, Brucella abortus, etc. Temperature. The optimum temperature for most of the pathogenic bacteria is 37 degrees Celsius. Again, it has of three types: psychrophiles, mesophiles, and thermophiles. Psychrophiles can withstand the temperature of 0 to 20 degrees Celsius. Most of the soil and water saprophytes belong to these groups. Mesophiles that have the temperature between 25 degree and 40 degrees Celsius. Most of the pathogenic bacteria belongs to this group. Thermophiles which can withstand the temperature of 55 to 80 degrees Celsius. Example, Bacillus stereothermophilus. pH. Most pathogenic bacteria grow between pH 7.2 and 7.6. Very few bacteria such as lactobacilli can grow at acidic pH below 4. Vibrio cholerae is an example of bacteria that can grow at an alkaline temperature and pH of 8.2 to 8.9. Light. Depending on the source of energy, bacteria make use of, and they may be classified as phototrophs and chemotrophs. Phototrophs. Bacteria deriving energy from sunlight. Bacteria deriving energy from chemical sources. Osmotic pressure. Microbes obtain all other nutrients in solution from surrounding water. Hence, factors such as osmotic pressure and salt concentration of the solution affects the growth of bacteria. Bacteria, by virtue of mechanical strength of their cell wall, are able to withstand a wide range of external osmotic variations. Organisms require high osmotic pressure are called as osmophilic bacteria. Sudden exposure of bacteria to hypotonic solution may cause osmotic withdrawal of water, leading to osmotic shrinkage of protoplasm, called plasmolysis. On the other hand, sudden transfer of bacteria from concentration solution to distilled water may cause excessive imbibition of water, leading to swelling and bursting of cells. It's also called as plasmolysis. The question for today's learning is what is called as mesophile and the temperature set for mesophile. Thank you, dear learners.